Can you hear me? Yeah, so today I'm going to go over what our current understanding of the gastrointestinal microbiome is from more of a perspective of microbiologists. So I do microbial physiology, ecology, and evolution for our field. So what we're going to do is we're going to go through, I'm going to introduce you to the main players of this game, who the microbes are, what they do. Then we're going to go in over what we kind of know currently about the gastrointestinal microbiome, and then move into some things which we're really excited about for future work, looking at like microbial activity, and then we're going to end with a little bit of discussion of some microbiome-related diseases. So Earth is basically a microbial world. Sorry, give me 1 second here. So this right here is a tree of life of all organisms on Earth, with our current understanding. The vast majority of diversity are bacteria, these groups right here. And humans are this little green branch here. Humans, trees, anything you can see, the macroscopic world areas are located down here. And this group right here is a group of organisms called archaea. These are little things that look like bacteria, but are very different from bacteria from the perspective of their evolution and their biochemistry. So in the human microbiome is composed majority of bacteria. There are a few archaea in there, and as well as some eukaryotes. And viruses play a key role in helping control the populations of these particular organisms within the gut. Within bacteria, what's really interesting, the diversity here is literally stunning. There are hundreds of what are called phyla of bacteria. These are really big groups of diversity. So like an example, one phyla of bacteria has more diversity than all of eukaryotes. So life on Earth really is microbial. And within the microbiome, we only see like maybe 5 or 7 different groups of phyla of bacteria which are found there. So there are hundreds of phyla of bacteria in the world. Only a few of them are actually found in the human gut. So what microbes do, which is interesting, they have effectively zero phenotypic diversity. They all look the same, the little squiggles and things like that, so they're not super interesting to look at, but they can do incredible metabolisms. So they can perform photosynthesis, they make all the oxygen on Earth that we breathe. They can perform aerobic respiration, which is something that we do. They can also perform different types of metabolisms called anaerobic respiration, where they can get energy from nitrogen compounds, sulfur compounds, iron, and a whole bunch of other different things. And then they also do something called fermentations, where they basically can do electron transfer between different types of organic molecules. And this is the main type of metabolism which is found in the gut. And for comparison, eukaryotes are phenotypically very diverse. Trees and dogs and whatever look incredibly different, but they all do the same thing. They basically do aerobic respiration, with the exception of a few things that make, a few plants which make oxygen. So how we tend to look at the human microbiome system, we look at it through the lens of microbial ecology. So it turns out in nature, microbes are found in every environment you can think of. On any surface, they're there. As long as there's water and an energy source, there's microbes. They thrive under a wide range of conditions. Here's some sampling sites that we work on in our other life, like hot springs at Yellowstone or deep sea thermal vents. And there's organisms which can live at pH 0 all the way up to pH 14, and organisms which basically can live at freezing temperatures all the way above the boiling temperature of water. These things are incredible. Microbes form complex communities which basically are structured by competition for resources, by mutualism, where they basically share compounds and help each other grow, by commensalism, which they just don't care if the other one's there, they're just doing their own thing, and then predation. There are some bacteria that go around and kill other bacteria and suck their guts out to make a living. Viruses do effectively the same thing, but they don't eat them. They just basically use them to make new viruses. And microbes play key roles in the evolution and maintenance of life on Earth. They literally produce our oxygen, they drive all of our biogeochemical systems, and they control the carbon cycle and everything. So now what we're going to do is go through, that's weird. I'm not sure where that I came from. It's an L, I see. So now we're going to go look at what our current understanding of the gastrointestinal microbiome is from the past like 15, 20 years of work within this field. So we do a quick brief overview of what we actually are looking at when we look at the human gastrointestinal tract. So this right here is a little diagram of the GI tract. We can see here there's an esophagus. When we process food in our mouth and it comes down through the esophagus into the stomach, through the duodenum, jejunum, ileum, and eventually through the colon, and then we excrete it. And all these different areas of the gastrointestinal tract have different functions, which are very important. One thing that's really interesting is the vast majority of nutrient absorption actually occurs in the small intestine. The large intestine, for the most part, its main function is waste storage, water absorption, and stool formation. One thing I want to point out that the amount of microbes which live in different areas of the gastrointestinal tract varies drastically. There's only a few microbes that can live in the stomach. It's very low pH. But then when we get into the duodenum and through the small intestine here, they have relatively low concentrations of microbes. When we see a large quantity of microbes is when we hit the actual colon towards the end of the gastrointestinal tract. And one thing I want to point out, so the vast majority of studies to date have been looking at characterizing microbes, who's there, and the majority of those samples are coming from stool. So we have to keep that in mind when we try to interpret what does this actually mean, because the reality is, the vast majority of the GI tract has very different function and very different type of microbes which live there. Besides physiological function in different areas of the gastrointestinal tract, we also see different types of disease states which are prevalent in different areas. And once again, if we want to study something like celiac disease, it's probably a good idea for us to get some other samples besides stool, because that mostly affects the small intestine. So this right here is a brief overview of what are the main kind of physiological characteristics within the gastrointestinal tract. In the small intestine, we see very fast transit time. So we eat food, it comes through quite quick. There's relatively low biomass of microbes, not a ton of them live there. It has a relatively thin mucosa layer here, where basically these polysaccharides which protect our own host cells from the microbiota. And what happens is mostly simple carbohydrates are processed there. So those are taken up first because they're easy to get at. And as we travel through the gastrointestinal tract, we see a slowing down, the transit time slows. We see a lot more biomass. We have a thicker mucosa layer there to help protect us from some of these microbes. And this is where the majority of complex carbohydrate processing occurs, like fiber, things like this. So if we actually want to study the microbiome, we have to actually measure something. And how we do this is microbiologists, is we take a look at what is an actual microbe made of. Microbes basically have nucleic acids. They have proteins which basically are things like enzymes which perform catalysis and structural features. They have lipids which basically form their bilayer and enclose the cell, and there's polysaccharides and a whole bunch of these small molecules which basically are parts of metabolism. This is what the cell makes when it's trying to do it, when it's doing its process of living. And to look at these different types of compounds, we have a number of different types of approaches. To look at the nucleic acids, we can do something very simple, something called 16S sequencing, where we just basically look at one gene in that organism that basically uses the marker to ask who are you. The 16S gene basically tells us what that organism is. We can also look at all of the DNA within an organism, something called genomics, and identify all the different genes that particular organism has. We can also look at transcriptomics, which is looking at the nucleic acid RNA. What that tells us is what genes are being expressed at a particular time. So what happens is DNA basically stores information and RNA processes it. So we want to turn something on and actually turn on a particular protein. We have to make that protein, RNA does that. If we want to look at which proteins are made, we can do something called proteomics, where we basically go through, use some really expensive fancy tools, and break down the proteins and then measure how much they weigh within a really fancy machine, and that tells us what protein that is. There are other things like lipidomics and things to look at sugars. And then also small molecules, we can look at those also with mass spectrometers. We actually can measure how much these things weigh in a depth. discovered so far. So the human microbiome consists of 10 to 13 cells. It's the same number of cells. It's basically the same number of cells that we have. There are thousands of species which can live in the gut. Majority of them fall into the category like Firmicutes bacteria and Actinobacteria. These are kind of high-level, coarse-grained groups of organisms. As I mentioned before, there's hundreds more of this type of thing. And when we look at these here, this right here is a phylogenetic tree of all the microbes which have been discovered in the in various human microbiomes to date within the gut. And the ones in blue here, the little blue ones, are ones which we can culture. So these are the ones that we kind of have more information about, people have been studying for years. But you can see the vast diversity of microbes in the human gut can't be cultured yet. We're still working on that. And so for the most part, we really don't know what a bunch of these things are actually doing very well. The gut microbiome is incredibly fascinating. There's at least 10 million genes that are encoded by all these different microbes, which is substantially more than we have. We only have around 20,000 genes. So the microbiome encodes orders of magnitude more diversity of genes than we actually have. One of the main problems with this is that we have no clue what most of them are doing. And it's really tricky and really difficult to actually figure out biochemically what these particular genes do. The average cell has around, an average microbial cell has around 2,500 genes. Most of these are differentially expressed. It turns out you only need about 500 genes to be alive. So what happens is they have all these genes, so when they see a new food source, they can turn those genes on to use that. Because you don't want to be wasting energy and turning those genes on all the time unless there's something there to do. So this is where we utilize things like transcriptomics, and looking at the genes is where genomics come into play. So one of the main take-home points from the Human Microbiome Project has been that it turns out there is a ton of diversity between individuals and their microbiomes. So if you look at two individuals, their skin microbiota look totally different from each other for the most part. Their GI microbiota look very different. So this makes it really tricky because when we're trying to understand like what diseases might be caused by what microbes, and we see such variation, we have to dig a lot deeper to try to understand what is causing that. So for humans, or for any animal for that matter, the microbiome, when we're first born, we have very little, our microbiome is very small. We have very few species. What happens is the microbiome develops over time, and it depends a lot on our environment. So things that actually lead to major differences in our endpoint are going to be whether our delivery mode at birth, how we were fed as children, what types of solid foods we had, and basically what our childhood diet is. So it turns out is we start with some groups of organisms, and then over time we start accumulating more and more diversity of microbes, and eventually we'll end up in different types of final states. Some of them which are quite different from each other, and depends on our food sources. Like with people with high fiber diets tend to have one type of microbes, and people with Western diets which have less complex fiber and more high fat and sugars tend to have a different type of microbiome. And that's something which is actually really important for human health as a whole. So when we go and actually look at microbial diversity in different human populations, we see that if you look at remote hunter-gatherer populations, one thing they have is really high microbial diversity of a whole bunch of different types of microbes which can live in their gut. And as we track through different types of living over through like farming or fishing populations and on into basically us, Western populations, we see a drastic decrease in microbial diversity. And this is probably going to be causing a large quantity of health issues for us. One of the other major discoveries from the Human Microbiome Project was that it turns out geography and ethnicity play a major role in variation as well. Different countries will be associated with different types of microbes, more prevalent than other countries. And here's a map, a recent map is showing a number of different types of variations between different areas of the world. And the color coding here is basically how urbanized these different areas are. So places in green have very low, have very low urbanization, things in like reds and yellows have high urbanization. So we see that actually this environmental cues actually play an important role in what our microbiome actually is. And also it turns out that our microbiome is not constant. It changes every day and it changes throughout our life. This right here is a study from 2 scientists out at MIT who basically sequenced their fecal microbiome every day for a year, just because they could. So, which is really interesting. So one of them, what we're seeing here, these are gross scale phyla level differences between these 2 individuals. You can see that they look quite different from each other already, even though they live really close to each other. So this particular person went to visit his wife's family in the Philippines, and for a while, and his microbiome changed drastically when he got there, because you're having different food sources and things like that. And when he returned, it looks like it's the same, but it really isn't, because it turns out that the level of discrimination between phyla there's a whole bunch of different species within each phyla. There's a lot of diversity. So what happened is, even though there might be roughly a constant amount of Firmicutes or like Bacteroidetes in his gut, they were different organisms that were found there afterwards. And this other person had a similar occurrence of dysfunction where he got food poisoning eating, and it completely changed his microbiome over as well. So as you can see, that throughout our life, our microbiome is constantly changing. So as I mentioned before, one of the things that was interesting about a lot of the previous studies is that these were mostly looking at addressing the question of who was there. And they did that usually mostly using stool, because that was easy to get, and it turns out that scientists have a hard time getting physicians to get them samples elsewhere. So when we look at, is this a good proxy for the whole gastrointestinal system? It turns out maybe not. So here's a study looking at monkeys, where it shows that when we look at the microbiome that is found in the lumen of the gut, which is like inside the tube, it looks relatively similar to stool. So the colon microbiome is relatively similar. However, when we look at the mucosa, which is a cellular layer that interacts with the human host, they were completely different species. And these other ones, they're going to be really important for interacting with the human host, and these are the ones which are going to be basically driving a lot of disease states. So we really have to understand what those microbes are and what they are actually doing there. So it turns out there really wasn't a whole lot of data done on this for humans. So Laura, she spoke before me, when she was fellow out at UCSD, decided to do a large-scale mapping of the human microbiome throughout the whole the GI tract. So she looked at 68 patients who basically came in for endoscopy. They ranged, they had quite a wide range between around 20 and 75. About half of them were female, and they came in for a number of different disease states. And what she did is she went through and took mucosal samples, which is the part that is important for looking at disease states, from the whole tract, from esophagus, stomach, small intestine, large intestine, using endoscopy procedures. And once she got those samples, we went through and started asking questions like, who's there? So we did some of these genome sequencing using 16S, asking what types of microbiome, what types of microbes are located in different regions of the gastrointestinal tract. And then she also looked at what types of metabolites, what kind of small molecules are also located in these regions, because this is really important for understanding interaction with the human host and asking what types of processing is done there. And so this is something which to date has not been done. So she was able to actually map organisms and the metabolites they're making throughout the whole gastrointestinal tract. So this right here is a picture that scientists love to show all the time, and it's super hard to read. So I'm going to tell you what this means. So basically, these are bar charts, which basically tell us the proportion of microbes in a given area are of what type of species. So the color here, so it looks like, let me turn it off very quickly. So the ones in blue here are Firmicutes. The ones in yellow are Bacteroidetes, and so on and so on. So what we can see here is at the beginning of the gastrointestinal tract, the esophagus, the stomach, and the duodenum, we see this is an area, by the way, that we talked about before. It has very low population numbers, not a whole lot of microbes there, but they're very diverse. As you can see, the little bar charts show a bunch of different colors. So there's a lot of different organisms which populate these areas of the gastrointestinal tract. And then once we actually hit the ileum, which is right here, it mostly evens out. So we see mostly blue and yellow. These patients right here had endoscopy because they came in with dysbiosis, they were sick. So this right here are mostly E. coli and salmonella infections. We can easily see them. They just like take over the whole GI tract. It's horrible. But for the most part, we see a large gross difference between the small intestine and the large intestine as far as like what types of organisms are there. We can also look at this using some weird math and statistics and ask ourselves, how can we visualize these communities and how they compare to each other by making these what they call principal component analysis plots. So I'm going to walk you through this and tell you what this means. So every dot is a single sample within her patients. And what we're looking at here, we're going to look at the large dots here are from the esophagus. So we can compare the communities. So basically, if the dot is close to each other, those communities are similar. And if they're far away from each other, they're quite different. So we can see here, once again, looking at the esophagus, the stomach with the larger dots here, and the duodenum, they're relatively close to each other. So they have similar community structures. Whereas when we get to the ileum and colon, they're quite far from each other, from the other ones. This right here is the ileum. Sorry, I'm not going to do this. And this right here is our colon samples. So that also looks different. So besides they have different species and they have different community compositions within the gastrointestinal tract. And this right here is a sample from one person where we were able to go through and do some special techniques called a double balloon endoscopy to get a long way through and get samples from that individual. And just, once again, you can see that the beginning of the gastrointestinal tract shows a lot of diversity, and then when we get to the large intestine, it looks quite similar. And we can see these communities are quite different. Like this is the esophagus and stomach, the duodenum are sort of close to each other, but they're quite far away from the colon ileum. So it tells us that there's quite a bit of diversity in where microbes are found and what they're probably doing there. And the important thing here is, here's his stool sample. So it's not close to anything else. So this is something we have to keep in mind when we're doing a lot of these studies, is that, and interpreting a lot of the previous studies, is that almost all previous studies were looking specifically at stool, and that might not tell us what we need to know about the microbial diversity. So besides looking at the organisms which are found there, Laura also went through asking like what kind of molecules are made. In the background here, you can see a biochemical metabolic chart. It looks really complicated because it is. Microbes can make a million different compounds. It's insane. And so what we want to do is ask what compounds are being made, and we would eventually like to figure out which organs are actually making them. So she went through, did something called untargeted metabolomics, where she takes her sample and puts it into the mass spec and see what comes out. One of the limitations of this particular technique is that it has very limited identification. So there's so much diversity in these molecules, we can only identify what 2 to 3% of them are, which is kind of sad. So there's a ton of work by chemists going on right now to try to increase that, but this is going to be a long-term problem. But the thing is that a lot of these small molecules have potential to, hold really important functional biological information or serve as biomarkers. So we really need to actually be able to do this in a way which will allow us to take advantage of that. So I'm just going to do a quick overview of like how they analyze the data. It doesn't really matter. Long story short, they do some fancy math and statistics on these, and are able to build these networks of molecules which are related to each other. Things that look similar, but they're not quite the same. They have like bells and whistles which are different. So this particular group is called a molecular network, and they share a couple things in common. This little tail here, this lipid tail on the end of all these different compounds are the same. So they are very similar, so they form this network which helps us identify what these other molecules might be, and so we can actually go through and try to use computational tools to predict what these are. This right here is some other little networks of things that she found in the stomach in different areas of the microbiome. And what we'll see is some compounds form large networks, which means that there are a lot of related molecules in these areas. Other ones are not so common. Once again, the problem is we don't know what the vast majority of these are. So we get large quantities of data, which is really hard to interpret. But we can do something similar where we look at how to, when we look at the microbiome, looking at how that community is structured. We can do the same thing by looking at the molecules, and we see something very similar. So molecules found in the esophagus are quite similar. Ones in the stomach are similar to each other, duodenum, ileum, and colon, which tells us we're actually seeing a real signal within these types of environments from looking at the small molecules. So the take-home point of this is we identified about 5,000 independent molecules in the study, and this right here are the number of molecules which we knew what they were, which is a very small number, turns out. And the ones in parentheses here are the ones which were unique to that region. So this is ways that groups are trying to start build up biomarker type studies. What we're looking for is what molecules are unique to different areas of the actual gastrointestinal tract. So this is a proof of principle study. I think it's the first one that was actually done like this, and there's a lot of work to be done in the future. So the take-home point from this is that the microbiome varies by region. Metabolome also varies by region. It turns out that stool and luminal aspirates might not be really good proxies for many disease processes. So we have a long ways to go in microbiome research, so we can start translating this knowledge into different types of healthcare. So one thing that we're also really excited about, this is something another project we're kind of working on right now, is when we look at most of the previous studies, we're just asking who's there. We don't know what they're actually doing for the most part. We don't know if they're active. So we don't know if they might just be hanging out there. A lot of times microbes just sit there forever. And the deep subsurface, they might sit there for 100 years or nothing, like literally. I have colleagues working this. So the thing is, we want to know which ones are active and which types of molecules are processing. So we're working with this really talented microbial ecologist at Montana State named Roland Hansen-Pickler. He's developing some of these new tools to actually ask these types of questions. What is actually happening right now for these microbes in where they're actually living? Usually what happens in microbiology studies is that environmental microbiologists, people who study things like Yellowstone and deep sea type vents, usually develop tools to understand this, and it usually takes at least a decade before those tools migrate to the human realm. So we're trying to do is shortcut that by decade. So these techniques are being developed as we speak. We're collaborating very closely with this group. We're trying to apply these directly to the human microbiome, so we don't have to wait that decade. So different ways of asking what's actually happening for microbes where they're actually living. As I mentioned before, we can do transcriptomics in an individual cell, looking at what genes are expressed, but we can also do called metatranscriptomics, where we actually look at the RNA of the whole community. So we're asking where are all the genes expressed in that community. And there's also really clever ways of using stable isotopes. These are just atoms which weigh slightly different. And we can use them to actually label things like DNA or different types of molecules, and then use fancy equipment to look at which ones were labeled. So there's one of these techniques is called Raman microspectroscopy. And sorry, to go faster. He's the only one in the country that does this. And so one of the things we're really excited about also is this new technology called BONCAT, which basically uses amino acid labeling for cells. So what this does is when cells are want to make new protein, they have to turn the genes on. So what we can do is we can give them, I didn't show it all. Sorry, something didn't work at all here. So I might have to skip this section because there's a whole bunch of stuff on the slides that actually are for some reason not showing up on this computer. But long story short, what we can do is we can actually use non-natural amino acids to basically tag proteins which are being made in real time. And then we can add a fluorophore to that tag and actually use them to label a population of cells with a special tag, which will look like the ones here in pink. And what that does, it tells us those are the actual cells which are actively utilizing a particular compound. And then we can actually sort them out into a special bin using a special cell sorter, and then ask what they're actually, who are they, and what are they actually doing. So what's exciting about this is what we can do is we can take a complex community and basically try thousands of different types of conditions all at the same time. we can ask which organisms can eat compound 1, compound 2, compound 975. All in parallel, label them, and then go do a large number of studies to actually characterize them, either using metagenomics, metatranscriptomics, and metaproteomics. So this gives us access to what organisms are doing live at that time. And one of the long-term goals is to be able to map that onto three-dimensional structures of communities, because communities aren't just this homogeneous mixture, they form these really complex communities where some cells are close to each other and there's different species in different concentrations, different places. So that's something that has to be done. So one thing that is something that people are really excited about is actually trying to do microbiome-wide association studies. So what this does is it links features of microbiome to specific clinical phenotypes. So we're looking at what are the microbes or metabolites or genes associated with a specific disease. And one of the problems with this is it requires very large cohorts to be statistically relevant. And basically this causes limitation on case control studies because it's really difficult to delineate cause and effect with these. Let's go through a couple of these. So these are some of the disease states which actually have been associated to date with the microbiome. So one of the exciting areas is looking at the microbiome associated with autism. So in previous studies, we've seen that basically there are clear differences in the microbiota in autism patients. However, the results are kind of contradictory. You know, one result says one study says one thing and another study says another thing. But since there is differences, people are really excited about actually moving forward with these studies. And a recent one, a study published very recently, was a study where they took 18 patients, basically getting antibiotics, bowel plants, and fecal microbiome transplant, and asked the question is, how are they doing after 2 years from this particular process? And it turns out this is incredibly interesting. It turns out their GI symptoms got substantially better, and so did their autism-related symptoms. And it turns out that was stable after 2 years, which is really interesting. So, and also the improvement in the GI symptoms and autism symptoms were correlated. And they also found some increases in bacterial diversity within these patients. So they got very excited about this. They're now doing a large-scale double-blind placebo-controlled study to see whether or not this is something that is going to be more general. And I'll end with a really quick overview of, one of the main picture goals of microbiome research is to actually do something with this, right? So we just don't want to have a whole bunch of pretty cool charts and figures and stuff. We want to be able to actually do something useful. So we want to be able to actually modify the microbiome in an intelligent way. Right now we have antibiotics. These are just like nuclear bombs that take out everybody. It's just a horrible thing. Other ways of modifying the microbiome are through diet modifications, but they're not very specific. It's very difficult to pinpoint things. There are probiotics which can be utilized, and two of the areas which are kind of exciting right now are fecal microbiota transplantations and phage therapy. Both of these, I think, hold a fair amount of promise. So fecal microbiome transplantation is basically taking a healthy stool from a healthy individual and transplanting it into an individual who's sick. And the idea is that hopefully those microbes will form a new community which can get rid of that particular disease state. And it's worked very well in C. diff infections. In fact, Laura was one of the first people in Utah to actually do one of these transplants. It was truly stunning to see how the person recovered in like literally 12 hours. It's bizarre. Looking at fecal microbiome transplants as being important is there was a really interesting study published recently. Well, it's not even published yet. It's online though, but that looked at what happens if we take antibiotics, kind of destroy the community. What's the best way of recovering that? And what they did is they looked at 3 different techniques. One using probiotics, one using fecal microbiome transplant, and one using using a spontaneous recovery. And how they looked at recovery, they looked at what the microbiome was over time and how the host transcriptome reacted to this. It turns out that basically the fastest recovery rate was through fecal microbiome transplantation or just waiting. And it turns out in this particular study, probiotics actually delayed the recovery of that particular community and by quite a bit. This is something that has to be replicated under different conditions, but it's something that's quite interesting. And then I'll end with 1 or 2 slides on phage therapy, which is basically, is there a way for us to pinpoint, target actually individual strains and take them out that are causing problems? And this is a development that started in Eastern Europe back in the 70s. They didn't use antibiotics, they used viruses to attack individual species of microbes and actually kill them. So there's a number of companies in the US right now working on this technology with the idea of maybe in the future we use a scalpel and take out the things we don't want and actually make this a little easier. One of the problems with that, when we look at this within mice, there are ways of actually taking a community, adding these viruses, and it knocks out the bad ones, like this right here attacking the orange ones and they're gone. This one attacked another group of microbes and they're also gone. However, what happens is when you take out one microorganism from this community, we see drastic changes in all the other ones. So their levels change quite a bit. And not just their levels of microorganisms, also metabolites. All these small molecules also are changing. So this is a very dynamic system, and it's going to be very difficult for us to do this in a way which is deterministic. So I want to end the basic mention that the human microbiome is really complex, and we're just starting to understand its structure, function, and dynamic. There's a number of very new, very exciting techniques which are coming out which will help us do this. And it seems like there's a lot of promise for microbiome application to actually for disease prevention and treatment in the future. And I'd like to thank Laura. I work with her at the University of Utah, our collaborator, Roland, at Montana State, who's developing all these tools. And then Laura worked with Rob and Peter Dorsey when she's at UCSD doing a lot of this microbiome mapping. And I can answer any questions.